Hello, everybody. Let's talk about amino acid. Let's learn about amino acid with their characteristics and the structure and the common and general functions. Please consider this an introduction about amino acids. Okay. First, let me share the screen now with you. Okay, working on the screen now here. Okay. Right. I think you can see the screen with a white background. So let's talk about amino acids. For the characteristics of amino acid, we will talk about nutritional needs or essentiality. We can classify amino acid based on that. Also, we can group the amino acid based on their metabolic rates. Right? So let's talk about those. There are 20, at least 20 amino acids that you can see as a part of protein. There will be a few more that we can talk about, but among those, some of them are considered or grouped as essential amino acids. If you look at the different papers or texts, you may see a different term called indispensable amino acids. Um, Actually, basically, those are same thing, but depending on the scientist or depending on the research group, you may see we use different term, essential amino acid or indispensable amino acids. In animal science, we are changing little bit to use this term indispensable amino acid rather than essential amino acid. But in this class, it's okay. We can use either one. Okay. So I'll rather use the older term called essential amino acid in this class. And some other amino acids are grouped as non-essential or dispensable amino acids. So that means some amino acids are nutritionally essential, we need to include those in the food or feed. And without eating or obtaining those amino acids, the animals or human will not be able to make a protein. So those are the essential amino acids. Whereas some other amino acids that those can be made or synthesized enough from different um, nutrients or the sources. So we don't have to obtain those amino acids from the food or feeds, and so we call them non-essential amino acids. Whereas sometimes there are few amino acids they becomes essential under certain condition. Whereas some other cases, some other conditions, they are not essential. So we call them, this few amino acid as together, we call them conditionally essential amino acid or conditionally indispensable amino acids. Right? So let's talk about those. And also, depending on how they are metabolized, and what are the end product of this amino acid metabolism, we can we can group amino acid as glucogenic or ketogenic. Right? The glucogenic amino acid, those are the one that will be converted into the glucose through gluconeogenesis. I hope you learn about that in other nutrition courses. And whereas there are few amino acids that are ketogenic, means they will be converted to ketone bodies through the acetyl-CoA. But there are some amino acids there metabolically, they can be either glucogenic or ketogenic. So we can group those amino acids into these three categories. All right, let me introduce the structure of amino acid. I am sure that you have this knowledge. You know about this from your undergraduate the biochem or organic chem. Okay? But let's just spend some time to get reminded here. Okay? Let's see here. This may be a very representing a structure of an amino acid. Always there is a carbon at the center. So we call that center carbon or alpha carbon. Okay, first one, yeah, alpha carbon, center carbon. When we draw a structure of an amino acid, we have an agreement that the center carbon at the center, alpha carbon at the center, but on the right side, this carbon will include a carboxyl group, COOH, or carboxylic acid group here on its right side. Whereas on the left side, we will have amino group or amine group, NH2. Okay? So this alpha carbon will have both carboxyl group and amine group. Upper side, it will have hydrogen. At the bottom, it will have R group or functional group. We'll call that R group in this class. So the center carbon or alpha carbon will have four compounds or groups attached to. However, if you look at this amino acid under physiological pH or neutral pH, it will have some modification. In the case of amine group, it will be ionized, becoming ammonium cation. So that is because this nitrogen will receive an additional hydrogen, and this amine group will be positively charged, so it's cation, so called the ammonium cation. Whereas this carboxylic acid group or carboxyl group will lose the hydrogen and it will be negatively charged. So it is carboxylate anion. So if you look at this one amino acid under at the neutral pH or physiological pH, it has a cation, but at the same time has an anion. Okay? So the compound or molecule containing positive charge and negative charge at the same time, we call them zwitterion. So amino acid is an zwitterion at neutral pH. Okay? Now let's talk about the R group or functional group, R group here. Depending on the R group, that will determine the different types of amino acid. Okay? The smallest amino acid is called glycine. Glycine here, okay? That's when the R group is simple hydrogen. But this R group is a hydrogen, we call that glycine. When R group contains a carbon, one carbon, of course, the hydrogen will be attached. It is called alanine. And there are additional carbons, two more, so total three, making the R group. We call that valine. When there's another one coming in the middle of this chain, the carbon chain, we call that leucine. And when the branch of this branch point here is changed here. We call that isomer of leucine, called in short is isoleucine. So if you look at these five amino acids that are introduced, glycine, alanine, valine, leucine, and isoleucine, their R group, it is mainly composed of carbon, is a hydrogen and carbon, is hydrocarbon chain. So if you look at this R group, it looks very similar to the hydrocarbon chain in a fatty acid. It looks like a little similar to that. they have some lipid-like characteristics. So we call them, as a group, we call them aliphatic amino acids. They are hydrophobic. If we look at the hydrophobicity of the amino acid, they are relatively hydrophobic to others, okay? To other amino acids. And now there are a few additional amino acids that I can introduce. In the R group has a carbon and hydroxyl group attached to that carbon. It is called serine. But then when there's an additional carbon attached to serine here, the carbon on the R group of the serine, it is called the threonine. So serine and threonine, it looks very similar, but depending on the additional carbon or not. And these 7 amino acids that I just introduced, their R group is not charged, and so they are together, they are neutral amino acids. Okay? The entire amino acid is not positively or negatively charged. So, and net zero, so it's neutral amino acids. Right? We already talked about 7 amino acids here. Okay? Let's move to the next slide. Here, please remember the structure, the basic structure of an amino acid here. Right now, I'm showing you 7 more here. The first 3. Now, the R group, when the R group is if the R group contains aromatic rings, like a phenyl ring, usually attached to carbon on R group, we call them together as aromatic amino acids. So because they have aromatic rings, like benzene or a phenyl ring here. Okay. So those are phenylalanine. It looks like alanine, but with the aromatic rings together. So we call it phenylalanine. Phenylalanine, okay? Alanine with a benzene ring here. When phenylalanine has an additional hydroxyl group, okay, at the end here, we called it tyrosine. When this aromatic ring it looks more complicated, but has an additional nitrogen here, make some kind of incomplete ring here, call that tryptophan. So now we have a phenylalanine, tyrosine, and tryptophan. Okay, and one more. I put this in a parenthesis because it can belong to other group. But histidine will also have a ring, but it is not an aromatic ring. It's called the imidazole ring. We will talk a lot about more about the histidine later about its R group, but its R group is not a complete aromatic ring, so it can. I would like to include histidine into another category, but I put that in the parentheses for under aromatic amino acid for now. Okay, so there are 3 amino acids belonging to aromatic group, aromatic amino acid. Those are phenylalanine, tyrosine, and tryptophan. Okay? Those amino acids are very strong hydrophobic and relatively stronger hydrophobic compared to other amino acids. And there are 2 amino acids very uniquely containing sulfur in their R group. So in the case of cysteine, its R group has a carbon and then the sulfur. I call that cysteine. Okay? And the amino acid with the longer chain, longer hydrocarbon chain with the sulfur in the middle, okay? We call that methionine. Okay? So the cysteine and methionine Those are the two sulfur-containing amino acid, or in short, sulfur amino acid, or SAA. And we have a proline. It looks very different from other amino acid because the structure itself, it makes a kind of ring or cyclic form. So we call that a cyclic amino acid, but proline looks like this, very different from the others. Okay. So now I added the 7 more, and we'll have the remaining here, right? Now, here, let me move this box here a little bit here. Okay. Now, you can see there are 3 amino acids, okay? Actually 4, including histidine, which has a basic structure, characteristics, or alkaline property. We call that a basic amino acid or alkaline amino acids. Those are lysine, arginine, histidine, and also I can include a special amino acid called ornithine. This ornithine would not be a part of protein, but it also has a amino acid structure, so I just include it here. But this ornithine, you can see the ornithine typically in the urea cycle in the liver. Right? If you look at the structure of R group, this amino acid, lysine, ornithine, arginine, and histidine, they commonly have nitrogen, additional nitrogen in the R group here. You have a R group of lysine, it has nitrogen at the end, which is NH2 amine group, amino group, okay? And ornithine also have the same. And arginine has actually 3 more here, and histidine has a nitrogen here too. So this amino group, additional amino group will, at the neutral pH, they will be positively charged, so they are cation. So it makes the amino acid alkaline under neutral pH. So we call them basic amino acid. So here, again, lysine, arginine, histidine, and ornithine. Now I can introduce the remaining 4 amino acids. They are a mixture of the acidic amino acid and amides. Okay? But if I introduce glutamic acid or glutamate, if it is in the salt form, or aspartic acid or aspartate, okay, these two, glutamic acid and aspartic acid, if you look at their R group, they commonly have an additional carboxyl group here. Okay. At the neutral pH, this carboxyl group will lose the hydrogen, will be negatively charged. So if you look at this one amino acid, glutamic acid, there'll be the net negatively charged. So that makes this amino acid acidic. So these two are acidic amino acid under neutral pH because they have an extra carboxylic group here and in their R group. When this glutamic acid and aspartic acid have a nitrogen attached to the carboxyl group on their R groups, they become the amides, and we call them glutamine and asparagine. Okay? So glutamic acid becomes converted to glutamine, and aspartic acid is converted to asparagine. Right? So all this together is a little more than 20, but these are the amino acids that we will be talking about in this semester. And please remember their properties or characteristics. I want you to know, okay, which amino acid are basic, acidic or neutral, aromatic, hydrophobic, and hydrophilic. Okay? If I add a few more things here, all the basic amino acid and acidic amino acid, they are strong hydrophilic. Okay? They're hydrophilic. Compared to aromatic amino acid, they're strong hydrophobic. They are all relative, relatively speaking here. Okay, so please know. So if you look at the um, protein, protein is made of a sequence of amino acid. If you look at this three-dimensional structure of the protein, this hydrophobic amino acid will tend to be in the core of this protein structure, whereas hydrophilic amino acid will stay outside. That's because the body, our body, is the water environment. Okay? All right. So we talked about how we classify amino acids. If I come back to essential versus non-essential amino acid, okay, I'd like to say there are here about 9 or 10, depending on the research group or textbook. But here I will say 9 of them are essential or indispensable. Okay? That means the animals or human will need to obtain these 9 or 10 amino acids from the food or feed. Okay? If we don't have enough of them, um, received from the food or feed, then we will have difficulties or making the proteins needed for the function of the bodies and tissue gain. Okay? So these essential amino acids are leucine, isoleucine, and valine. Okay? Those are neutral amino acids. Lysine and histidine, those are basic amino acid. And threonine, okay, neutral amino acid, methionine, and sulfur amino acid, tryptophan, and phenylalanine, those are aromatic amino acid. Those are 9 of them. If you look at some other text or research group, you may see that arginine is also included as an essential amino acid, but still I can consider it as a conditional, so we'll say a 9 essential amino acid here. And I put some term called here BCAA. BCAA means in full is a branched chain amino acid. The R group has a branch point. So R group is made of carbons, but they have a branch. If you look at the structure of the R group, those are leucine, isoleucine, and valine. So we call them together as BCAA, branched chain amino acid. Also remember this methionine is sulfur amino acid or sulfur containing amino acids. Now, compared to essential amino acid, I can group this, at least 5 of them, as conditionally essential. So again, some other research group may say the arginine is essential, but in this class and then the papers and texts that I will use will consider arginine as conditionally essential. So it is arguable and depending on how you look at the arginine. Okay? When we say conditionally essential, and these amino acid becomes essential amino acid under specific condition. Okay? In the case of arginine, it becomes essential for the infants and pregnant animals or pregnant human during early lactation, and also for poultry, avian species, arginine is essential. Okay? Because during that time, even though arginine is made in the animal body from kidney, Okay, but even though it is made, the needs for this arginine becomes a lot more increased for the infants and pregnant and lactating animals and poultry. So that's why it becomes essential. The glycine and proline, proline and glycine are known to be nutritionally essential for the growing chickens because even though proline and glycine is made in the body of chickens, their needs for growth, their needs of proline and glycine for protein synthesis becomes a lot greater than what chickens can make in the body. Right? So in the case of proline, if I summarized findings from many research papers, in the case of chickens, the de novo synthesis of proline meets about 90% of the requirements. Okay, so you need additional proline through the feed for chickens. The case of glycine, de novo synthesis glycine meets about 60 to 70% of the requirement, also meaning that glycine need to be supplied from the food and feeds. Okay, there's another new amino acid I show you here is taurine. Okay, taurine can be synthesized from the cysteine. Okay, but in the case of feline, like cats, they don't have a very specific enzyme producing the taurine from cysteine, and so there's lack of de novo synthesis of taurine for the feline. Right? So for the cats, if you look at the cat food, we should make sure that taurine is sufficiently provided. Right? But this taurine is also can be obtained from the animal tissue. Okay? So this feline, which is carnivore, naturally they will get the taurine from their food, which is meat. Right? But however, if you're feeding your cat uh, of the feed based on the grains, well, without sufficient animal tissue, then probably taurine will be deficient in that cat food. So most likely, and they will have a supplemental taurine. The taurine is supplemented. Okay. The last one is glutamine. Get converted from glutamic acid by having additional nitrogen to this extra carboxyl group in the R group, glutamine. Okay, this is an amino acid is well utilized as an energy source very well by the immune cells and also the enterocytes in the um, intestine. Okay? So when do we need the immune cells to be activated and functions is when we are sick, when animals are sick, right? So the glutamine becomes essential because the immune cells will consume those, will utilize glutamine and becomes insufficient. So for the sick animals, we will see that the needs for glutamine increase and thus it becomes essential. So briefly, we talked about conditionally essential amino acid, but later when we talk about individual amino acid for their metabolism, and we'll talk about this in further details. Okay? Before I move to non-essential amino acid, I'd like to introduce Professor David H. Baker. Okay? This is a picture of Dr. Baker. He deceased in 2009, but in amino acid nutrition, regardless of human or animal, animal means including all livestock or companion animals. Dr. David Baker is a highly respected big name in amino acid nutrition here. Um, when I was student, everyone knew his research, but I don't know today um, because his papers is kind of rather little becomes old. If you simply check the Google, maybe you don't see Dr. Baker's papers coming up on the first page. So these new students, young students these days often do not see his name. But please remember, Dr. Baker has so many research papers with originality. So his original research papers are from many of them are from Dr. Baker's and his group. Okay, so I'd like to introduce him here. Okay, I was fortunate to have him as my PhD committee when I was a student at the University of Illinois. Okay? This is a one paper that I posted on the Moodle for you to read. It is very old paper. However, this is very classical and very original research papers showing why glycine and proline is essential for growing chickens. So I'm sure you're a graduate student. I'm sure that you read so many research papers and review papers, and please add this too. And this will give you a very good fundamental basis about the nutrition research. Right, I made a one-page summary, but I will not read this because I want you to read the paper. Okay, please read it really. Okay, don't put this paper into the AI asking the AI to make a summary for you. Please don't do that. Okay, please read this entire paper and compare. What you learn is similar to my own summary here. Okay? It is about why proline becomes essential and why glycine becomes essential from the animal feeding studies. Okay. I'd like to introduce another scientist, great scientist here. This is Dr. Guo Yao Wu. Yeah, this is his name here, Guo Yao Wu. He was a distinguished professor and a university fellow at Texas A&M University. Um, he's just very recently retired in 2025, so he is not longer with Texas A&M anymore, but his research is always being cited and his papers are always read, especially in amino acid nutrition metabolism related to arginine, glutamine, all methionine, and so on. Okay, so please remember his name here. In one like to introduce one of his review paper. I was fortunate to be a co-author of this paper, but this is one of the very highly cited papers that cited by other peers almost 2,000 times. And if you want to know about details about the arginine metabolism and why it becomes essential, under what condition, this is very good paper to read. So I also posted this paper, review paper, for you to read. So please have a time. It may take maybe a few hours for you to read and follow. Okay, maybe a little hard to understand everything here, but help you see, follow the main story here, telling why arginine becomes essential. Okay, so I provide a summary here. Very briefly, um, you see the arginine in the urea cycle in the liver. So in the hepatic urea cycle, you see that arginine is synthesized, okay, from argininosuccinate. However, please don't please know that arginine from urea cycle will not be exported to outside. This arginine in the urea cycle is trapped in the urea cycle because arginine will be very efficiently converted to ornithine as it provide urea. Okay? So the arginine in the urea cycle will not provide arginine to the body. So that's one misconception that people may have of saying, oh, arginine is made all the time in the urea cycle, so it is not essential. But that arginine is not useful for the other tissues. Whereas the arginine is made from kidney. Actually, kidney is the main tissue in the body providing arginine. Okay? This kidney will utilize the citrulline, which is exported from the small intestine. The enterocytes in the small intestine will make a citrulline using glutamate, glutamate, and proline, and the citrulline will be excreted and transferred to kidney, and kidney will convert the citrulline to arginine. And this arginine is available to other tissues. Okay? And so in that way, arginine is not essential because the kidney will supply arginine. But in the case of neonates and poultry and feline, arginine becomes essential because the neonates, they have inefficient, they don't have efficient production of arginine. And same for the chickens and cats, they are lacking specific enzymes for the production of citrulline in the small intestine. Okay. Again, this is summary. We'll also spend time just learning about arginine later in this semester. And now the non-essential amino acids. Okay, those are cysteine, tyrosine, glutamic acid, serine, alanine, aspartic acid, asparagine. Those are synthesized in the animal cell just sufficiently than what we need for the body function and making proteins. Okay? And also at the same time, the most typical food and animal feeds will have ample amount of these amino acids, and we are not considering supplementing these amino acids in the feed, which is correct until most recently. However, the world is changing so quick, and if you have heard about some term called low protein formulation, that doesn't apply to human nutrition too much, but in animal nutrition area, making a feed with very low crude protein to help the to reduce the nitrogen excretion through manure. In that kind of application in animal feeding, you may see sometimes you may see the needs of supplementing these non-essential amino acid in the feed, right? So all these amino acid I just mentioned here, all of them, you can buy. You can buy from a manufacturer, depending on which amino acid can be very inexpensive or can be very expensive. As an example, one of the essential amino acid. So since it's essential, there are good production of this amino acid, lysine. Lysine, which is basic amino acid, this lysine, the production of lysine globally is pretty good. And it is only 80 cents or 90 cents per kilogram. Pretty inexpensive. Whereas, which amino acid? I can say here, like a serine, there's no big market for serine, but still serine is made for pharmaceutical area or medical area. In the case of serine, you can buy serine from Ajinomoto plant in Raleigh, North Carolina. It will be around $30 per kilogram. Compared to lysine, 80 cents, serine, $30. The big difference, right? So, but you can buy. Now, let's talk about functions of amino acid. Okay? Okay, moving from characteristics and functions. I think depending on what happens to those amino acids when they're metabolized or catabolized, we can group them into different categories based on their It's the functions. Right? Some amino acid will be grouped as a glucogenic amino acid because when they're catabolized or oxidized, they're converted. Eventually, they're converted into glucose through gluconeogenesis. Whereas a few others are ketogenic amino acids because when they're catabolized or metabolized, they will be converted, eventually converted to ketone bodies as they go through the acetyl CoA. About five of them are they can be either glucogenic and or ketogenic. So they have a dual properties. So they can be they become both glucose or ketone bodies. So if I show you what they are, okay. Now there's 20 some amino acids are grouped by their fates. Okay, glucogenic amino acid, arginine, methionine, valine, histidine, cysteine, glutamic acid, glutamine, aspartic acid, asparagine, glycine, serine, proline, alanine. So many. How do I know? So many. Yeah. Whereas the ketogenic amino acid, there are only two. Only two are true ketogenic amino acid. They become ketone bodies when they're metabolized. Those are leucine and lysine. Leucine and lysine. Oh, well, there's something interesting. It's not academic at all here, but I'm just telling you, out of 20 some amino acid, there are only two amino acids start with the letter L. Okay, those are leucine and lysine. But somehow these two amino acids starting with the letter L, they are ketogenic amino acids. Oh, easy to kind of remember. Right? There are five amino acids that are both glucogenic and ketogenic. Right? Those are tyrosine, tryptophan, threonine, and isoleucine, phenylalanine. Okay. This is not academic at all. Another stuff that I'd like to just tell you as a joke. Okay. There are only three amino acids starting with the letter T: tyrosine, tryptophan, threonine. All those three are both glucogenic and ketogenic. Oh, interesting. And then two more: isoleucine and phenylalanine. Okay. Is there any amino acid starting with I other than isoleucine? Okay, there's only one. Isoleucine is both glucogenic, ketogenic. And then you add a phenylalanine here. So we want to know, okay? We want to know which amino acid becomes glucose or ketone bodies or both. Okay, this is the fundamental basics about amino acid metabolism. This is the introduction, but we will need to know about those. Okay, it's a little memorization at this moment, but later when we talk about the metabolic pathways of each amino acid, we will understand why they're ketogenic or why they're glucogenic. So please wait a little bit, but for now, we need a little time for remembering this. Now I'd like to introduce the term called the functional amino acid. It's nothing related to essential T. So we're not talking about essential or non-essential amino acid, but we are grouping a few amino acid with a very special function when they're converted into some other compounds in the body, and they have very essential functions in the body. So we call them, we will group them as a functional amino acid. Right? In animal science, we started using this term, functional amino acid, since 2007. I don't want to be arrogant here at all, but this is as far as I know, we were the first research group to coin this term functional amino acid in animal science. I don't know about human nutrition, but in animal science, the first papers using this term was 2007. And we also include this term in the Encyclopedia of Animal Science in 2007. But the functional amino acids, there are like 5 to 7, or it could be more. I like to introduce them. By the way, if you find any papers using this term, functional amino acid, prior to 2007, in human nutrition especially, if you let me know, that'll be great. I'll be very curious. No, now, I'd like to introduce just 5 of them. There will be more, but these are the well investigated functional amino acid that we know in animal science. Now, arginine, methionine, tryptophan, and phenylalanine, and glutamine. So because of their functionality, you can also buy these amino acids from the manufacturer, and they becomes more affordable to be added in animal feed. As an example of arginine, prior to 2007, it was about $20 per kilogram, but now it will be like $5 or less per kilogram. The price is dropping as more manufacturers are making more arginines. All these amino acids mainly made from the bacterial fermentation, okay, using the bacteria called Corynebacterium glutamicum, okay? Only except for the methionine. We'll talk about that later too. The case of arginine, when it is converted to ornithine, it will produce a nitric oxide, NO, nitric oxide, and this nitric oxide will be used to produce the cyclic GMP, and that is related. If that happens in the endothelial cell, making the blood vessels, that will be the cause of relaxation of the muscle. So it is improving the blood flow, causing the vasodilation. It is a well-known mechanism. So arginine can be used for that in together with the nitric oxide donors. And also, arginine is known to be converted to creatine. It is related to providing energy in the muscle. And also, arginine is converted to polyamines that we also use for the cell proliferations, including the blood vessels, making the blood vessels and so on. The case of methionine, through it becomes a cysteine and also further converted to taurine, so it can be a source of taurine, and also it's converted to polyamines such as spermidine and spermine, and also methionine will be converted to cysteine and further converted to glutathione. Okay? Glutathione is one of the most effective antioxidants in an animal cell. So it has functions of through this compound. The case of tryptophan, okay, it is very famous because it is a precursor of melatonin and further to serotonin and further to melatonin. If this happens all the time in the intestine related to the muscle contraction in the intestine. Also, surely it happens in the hypothalamus in the brain, providing the serotonin is a neurotransmitter related to the changes of the mood and so on. Melatonin also related to circadian rhythm and causing the sleepiness and so on. And even though it is not efficient at all, tryptophan can be converted to niacin. Niacin is a B vitamin. So the niacin can be obtained from tryptophan, but very poorly efficient. Yeah, 30-to-1 ratio. Okay. Phenylalanine. Okay, phenylalanine is another interesting amino acid with very special functions when it is converted to melanin in the melanocytes, in the cells on the skin. Okay, melanin will cause the pigmentation. Also, phenylalanine can be converted to dopamine, neurotransmitter, followed to epinephrine. Because of that, I mean, phenylalanine is very interesting, it can be interesting amino acids. But for with some genetic defects, some human will have a problem with catabolizing the phenylalanine, becomes very toxic. So for certain group of people, phenylalanine should not be taken. And do you know where you can find phenylalanine in the food and beverages? Of course, phenylalanine is abundant in the protein, animal meats, or even the vegetable proteins. You find the phenylalanine, of course. Uh, However, you know this aspartame, the sweetener. This aspartame will include a phenylalanine. So if you drink Coca-Cola Zero, Coke Zero, it includes aspartame, which contains phenylalanine. So this kind of beverages will indicate, okay, this can will include phenylalanine. So be careful if you cannot metabolize phenylalanine. Case of glutamine. By the way, because of the some people worry about that aspartame, if you consume so much, is carcinogen. So the consumption of aspartame becomes reduced. So there are a lot more phenylalanine available on market in these days. It's interesting things going on. Glutamine, this amino acid can be converted to glucosamine, which is helping the joints health. And also, immune cells will eat this glutamine as an energy source and causing the activation of immune cells. So these are the example of 5 amino acid, very important functions when they are metabolized into other compounds. But again, during the semester, we will talk about each of them. But before we finish, I'd like to just show you one paper. It's about the arginine. Arginine, because I spent years of my research time related to arginine, and this is just one of the example of the research. This was collaboration with my colleagues in China Agricultural University in Beijing, China. We fed newly weaned young pigs. They have a limitation of making arginine in the body, but we supplemented their feed by adding 0.7% of L-arginine. So it's a arginine supplemented diet. We fed them, and we sampled their small intestine at 28 days after feeding, and we looked at the structure of villi in the duodenum and jejunum. You can see that the pigs with the diet, eating arginine supplemented diet, they maintained higher villi, okay? Meaning their villi are less damaged, destroyed, or their villi has been grown up, repaired very efficiently. So they maintain around 400 micrometer, whereas the other pigs without supplemental arginine, their villi was very short, okay? Which is very common for these newly weaned pigs. When we looked at this vascular endothelial growth factor in the epithelium, in the enterocyte, the tissue in the duodenum and the jejunum, I should say mucosal tissue, you can see that this tissue will express this growth factor highly when they are consuming, from the pigs consuming the arginine supplemented diets, meaning the arginine was effectively providing nitric oxide and causing the growth of nitric oxide and polyamines and causing the developing the blood vessels in the small intestine. Okay? So this is one example of research showing the functionality of arginine when arginine is fed to the animals that conditionally requiring the supplemental arginine. Now, last slide for this section, I'd like to show you very general functions of amino acid, regardless of the individual amino acid. It's very general for all amino acids. Okay, most likely, this 20 amino acid that we're talking about here, they will be they will be used in protein synthesis. So that's one of the most important functions and use of amino acid. They will become a protein. Okay? And when there are excess amino acid in the body, it will be catabolized and to glucose or ketone bodies, and they will be used for generating energy. And when the amino acids are moving in the body or converted to other compounds, it will give or donates the carbon and nitrogen. So actually they are transferring carbon and nitrogen in the body. As a form of polyamines, they are carrying the nitrogens as well. There will be becomes other compounds. So some amino acids are precursors to other compounds like neurotransmitters like tryptophan or phenylalanine and so on. And then to other nitrogen-containing compounds like methionine and arginine becomes polyamines. And it helps the renal regulation of acid-base balance because amino acids are zero ions containing acidic and basic properties, and certain amino acids are more stronger acidic or alkaline. And they will also, when they work in the signaling in the body as well. So these are very general functions of all the amino acid, most amino acid that commonly possessing. Okay, so I'd like to conclude this section talking about the characteristics of amino acids and the functions of amino acids. So this is an introduction of all amino acids together. Right? Thank you, and I will stop sharing the slide and coming back here. Okay, so I hope you can read these actually 3 papers that I mentioned in this lecture, and I posted them on the Moodle as well. Okay. Also, if you have any questions from this section, you please free to contact me or post your questions on the Moodle. Okay, the course website actually. Okay. Thank you.